In this video, we will discuss about the interaction of the cells in TME, and in terms of the ferroptosis, we will discuss the interactions of the cells within the TME. So these are the cancerous tissues and the neutrophils that are uh, coming towards the cancerous tissues because of the activation of the immune system. So our starting point of this this figure is from this ferroptotic cancerous cells. So ferroptotic cancerous cells will release some of the DAMs. And how these ferroptotic cancerous cells occurs? These cancerous cells are being recognized by the immune system, and they will recruit the neutrophils, and these neutrophils that is will release some cytokines that will trigger the ferroptosis of the cancerous cells, and this ferroptosis of the cancerous cells will release some of the DAMs, and that will further maturate and activate the antigen dendritic cells into the antigen presenting cells, and that will trigger the immune response. So this is how the cancerous cells get ferroptotic, and after this cancerous cells get ferroptotic, it will release some of the DAMs and also some of the other cytokines such as this HMGB1 and ATP. And the HMGB1 and ATP, the function of this is to function of this is to is related to the cysteine that is necessary for this GCF production. So for example, here it is shown that this HMGB1 is been recruited by this tumor associated necrophils, tumor associated neutrophils, and that will help in this that will help in the this cancer tissue attacked by this tumor associated neutrophils. And simultaneously what will happens is that these cancerous cells will release this some of the cytokines such as this GCSF and TGF beta also. So the GCHF, both of these functions is that it will convert these neutrophils into this N2 phenotype, or the tumor associated neutrophils, and its function is to prevent the immune response and act as the immunosuppressor. So the neutrophils, that's actual function is to kill the is to have the pro-tumorigenic anti-tumorigenic response will starts to act like starts to produce this uh, immunosuppressive response because of the changing phenotype into this N2 uh, neutrophils, and this is occurs because of the cytokines that are released by cancerous tissues such as this GCSF and GCSF. GCSF are the byproduct of this ferroptotic cancerous cells. So along with this HGMB will also helps in this change in the phenotype of these neutrophils into this N2 phase. And let's also discuss the macrophage kind of phenotypes. The monocytes are also a part of the immune response, and that are also recruited at the site of the cancerous cells. And how they are how they functions is that the ferroptotic cells exosomes are been are been act upon this cancerous tissue. The exosomes that has these cross mutations and that are released by the ferroptotic cells are polarized on the tumor associated macrophage that are act upon the cancerous tissue. Along with this, the ferroptotic will also release this same OOH, and it will provide a signals like eat me signals. So they will polarize the tumor associated macrophage to act upon the cancerous cells. However, the because of the presence of these cytokines and also the exosomes polarization, these monocytes are also differentiated into this M2 type macrophages or tumor associated macrophages, and its function is also to have the immunosuppressive effect. So what will happen is the dams and other cytokines that are released by the ferroptotic cells because of the immune response will ultimately help in suppressing the immune response, uh, such as this neutrophils are converted into the N2 type uh, neutrophils or tumor associated neutrophils, and also the monocytes are converted into the M2 type macrophage or tumor associated macrophages. So both of them has immunosuppressive effect. So this is how the ferroptotic cells uh, the ferroptotic of the cancerous cells will not have a long lasting effect, but they will finally leads to the immunosuppressive effects. So it is like a feedback loop of the cancerous cells. That is, if there are too much ferroptosis occurs, then it may cause immunosuppressive environment. That will prevent the further ferroptosis or other immune response against the cancerous cells. So I hope you got an idea about the dual response of the ferroptotic cells that we have also discussed in one of our previous videos. If you need to understand about this dual role of these ferroptotic cancerous cells, you can go through the reference that has been provided in the description, and also you can refer to our previous videos. Those link uh, can be found from the I button. So that's all for this video. Thank you for your time.